And now moving on to some more genotoxicology. So the first one is the Comet Assay, a matter of heads and tails. It will be given by Amaya Sketa from the University of Navarra.
Thank you. So I have prepared a few slides explaining how the DNA is and things related to the DNA. I'm not sure if this is needed or not, so I'm going to check first. What is the DNA? What is the DNA? One, yes?
Assembly of oxidative bases.
Sorry, again?
Assembly of oxidative bases.
Where are you up? Well, one more try. Okay, where is the DNA? Let's start. Do we have DNA? The animals have DNA?
Yes.
Yeah. Where is our DNA? In the nucleus of the cells. In which cells? All of them. All of them. Well, mostly all of them, but yeah, all of them. Okay. How is the chemical composition of the DNA? Do you know that? What do we have in the DNA? We have some answers here. It says we have nucleotides. Yeah. Okay. Some more. Those are sugar. Yeah. So each nucleotide has a sugar, has a phosphate group, and has a nitrogenous base. Yeah. It's a polymer. Okay. This is the monomer. So this is repeated a lot of time. But we have 4 different types of bases. Yeah, are you familiar with that? We can have purine bases, which are thymine and cytosine, and we have pyrimidine bases that are adenine and guanine. Are you agree with that? Yes. Okay. Then these monomers are linked to the others with bonds between the phosphate and the sugar. But we have like a double. Okay, people are saying yes, yes. Now it's okay. Now it's coming. And then the bases are linked through hydrogen bonds. And we have complementary bases. Yeah, so guanine is always with cytosine, thymine is always with adenine. And it is twist. Okay. Yeah. Are you okay with that? So yeah. I may lie at some point. So be ready. And then it's packed. Yeah, there are different at different levels. It's compacted. You know how long is our DNA? Kilometers? No, one cell. 2 meters. So you have to pack 2 meters of something, of a chain, in the nucleus of the DNA. So it's extremely compacted. One thing I want to clarify, well, this is the different levels of compactation. When the DNA is most compacted is when they form chromosomes. But it is only happening in the mitosis, when the cell is going to divide. Meanwhile, the DNA is not forming chromosomes. So we can see it like that. Okay, this is a karyotype with different chromosomes, but in fact, when the cell is in the interface, it is like that. Okay. Okay. Good. Why do we have DNA? Why do we need DNA? Why all our cells need DNA? We can read it. Yeah. They are making proteins. Okay. So the DNA is transcribed to RNA, is the same, but with a different sugar in the nucleus. Then the DNA goes to the cytoplasm, and there is translated to proteins. Are you happy with that? You will have a mutation in the DNA. We will have a protein that is not working, or is working, but not the same as it should be. Maybe slower. We are thinking about an enzyme. Okay. Okay, a bit more like before explaining you what is how to perform the Comet Assay and the principle of the Comet Assay. I need you to know this, I mean, all of this about the DNA, but also something else. Okay, the DNA is in the nucleus and is very protected by two membranes, apart from other components. We have the membrane of the nucleus and we have the membrane of the cytoplasm, because it is very important for the cell to maintain it. But sometimes it can suffer different attacks from chemicals that come from outside of the cell, or even for compounds that are produced by the cells. Okay, so the DNA here, we can see different lesions that can appear in our DNA. We have breaks. We can have single-strand breaks. There's a break in one of the strands. Remember, we have two. Okay, we can have double-strand breaks. We have the breaks in the two of them. The bases can be oxidized. It can also be alkylated. We can have chemical compounds that attach covalently sometimes to the DNA. So we have bulky adducts in our DNA. We have compounds that can produce intra-strand crosslinks. We have compounds that can produce inter-strand crosslinks, so it can link the bases. Yeah, okay. We can also have some adducts that are in the backbone. You can see it. We can have even proteins that link to the DNA. So we can have all of these lesions. But these lesions can be repaired. And then after, if the repair is properly carried out, we will have a healthy cell again. If they are not repaired or they are misrepaired, normally the cell notices it and then goes into apoptosis. Do you know what is apoptosis? Yeah? Everyone knows what is apoptosis? Okay. If it doesn't go to apoptosis and the lesion is there when the cell is going to replicate, the cell can produce cells with mutations. So the information has changed. So if the information has changed, we may have a protein that's not working properly or it's not working at all. So if we have mutations, so it's non-repair or misrepair, and we have mutations, then of course can go to apoptosis again. So the cell cannot sit and go to apoptosis. Or we can produce proteins that are not working properly or are not working at all. And these proteins, depending on their activity, can produce cancer or other diseases. Okay? Mutations, we have two types. Point mutations, when it occurs in a small amount of bases, or chromosome aberrations. Chromosome aberration, I've put them in italics, because they can come from these pre-mutagenic lesions, or they can come from another processes, like problems with when the chromosomes are going to be segregated between the two the daughter cells. Okay? Good. What we are going to measure with the Comet Assay are these lesions. So they are lesions that can be repaired. We have other assays to detect mutations. Okay, I don't know if you are going to learn about them Ames test, mouse lymphoma assay, transgenic rodent gene mutation assay, micronuclei assay, chromosome aberration, mouse lymphoma assay. Some of these assays can be applied in vitro, some others in vivo, some others in humans. So some can be used in a human biomonitoring study, some others cannot. We are going to focus on the Comet assay today. So at the beginning of this year, we published this paper in Nature Protocols, and the title is Measuring DNA Modifications with the Comet Assay, a compendium of protocols. It means that there are many different protocols to perform the Comet Assay. And one of the reasons is because you can apply to many different types of samples. So here we have in vitro. Do you know what is an in vitro assay? Yeah, okay. So I can apply in vitro in cell lines, 3D models, zebrafish embryo, that also considered in vitro assay, yeast, fungi. It can also be applied in vivo in plants, as we have seen in the previous presentation. It can be applied in non-mammalian models, and here we have crustaceans, insects, mollusks, fish, etc., etc. It can also apply in mammals, including laboratory, domestic animals, wildlife animals, and also human. So it's really, you can apply to every tissue or sample from which you can get a cell suspension. So this makes the assay very, very powerful. It is used in human biomonitoring. It is used in ecological biomonitoring. It is used in genotoxicity testing, which means if we want to know if a compound is genotoxic, we can also use this assay. And of course, in basic research, if you want to, I don't know, to investigate something related with DNA repair. Or... Okay. So there are different, as I mentioned here, compendium of protocols. There are different protocols. We are going to start with the simplest one, which is the standard Comet assay, which is used to detect single-strand breaks and double-strand breaks. Please interrupt me whenever there is something that you don't understand, okay? So as I mentioned, we have to start with the cell suspension. And the cell suspension, okay, here we have this is cells in culture or blood. Here we have whole organisms. We can smash it and prepare a cell suspension from it. Or we can have tissues. We remove the tissues and you prepare a cell suspension from them. I'm not going to explain how to prepare the cell suspension because it differs a lot from sample to sample. Then you collect the isolated cells and they should be put it on ice. Why do you think they should be put it on ice? Okay, this is one point. There could be proteins that can degrade the DNA. So in that way, you stopped that proteins. Another reason, this is one of the reasons, but there is another one.
It's very stable and you put it on ice in general, like you protect it from additional damage that is not from your exposure.
Could be, yeah. We can add it as a third one. But there is one missing. DNA repair. We don't want the DNA repair to occur. We want to know if we have certain lesions there. Okay, that's why we put them on ice. Okay, this is the second step is to embed the cells in agarose. Normally it's used 1%, but it could be another proportion, but in agarose on microscope slides. Do you know what is a microscope slide? This is a microscope slide. Can you see it? Yeah, okay. So what we are going to do is we have the cells and we have agarose at 37 degrees to have it melt. We mix them in a tube and then we put 2 drops in the slides. And we create 2 small gels. So those small gels contains a lot of cells inside. So the cells are stopped in the gel. Okay? We have our 1,000 cells in each of these small gels. After that, we are going to lyse the cells. And to do that, we put all the slides in a lysis solution. The lysis solution contains high concentration of salts that will produce an osmotic attack, okay, a shock. It also has detergents to destabilize the membranes and has high pH also. It helps, let's say, okay. So what we are going to do with the lysis solution is to convert the cells into, not to convert, is to remove membranes, remove proteins, remove even some of the histones. We didn't talk about the histones when we were talking about the DNA. Yeah, for the compactation, they use histones, which are like big proteins, yeah, okay. So it also removes the histones. So what we have at the end after the lysis is a nucleoid, which is supercoiled DNA that is attached to a nuclear matrix at intervals. So it's, sorry, look at me, it's something like that. You have, like, this is like the nucleus, and then we have the DNA, like a loops all around. Yeah, okay, this is the nucleoid. Okay. So we have that. We have the nucleoid. So now here, sorry, here, instead of having the cells, we have the nucleoids. The fourth step is alkaline treatment, which is done at pH higher than 13, and the electrophoresis. So during the alkaline treatment, we are going to break all the hydrogen bonds between the bases. Do you remember that the bases were, yeah? You said, yeah, okay. So we are going to break all these hydrogen bonds. Then the next step, the electrophoresis, is the tricky step of the assay. We can say it's the most important step. Yeah. Why do you need it? Yeah. Well, in fact, you don't need it. You will see later why. Okay, you can do it without, and see you will see some. You will see the effect of having breaks in the DNA. It makes the assay a bit more sensitive. Yeah, I will explain you why later. So then, well, sorry, I didn't mention that this is an electrophoresis tank. Well, it's meant to be an electrophoresis tank, okay? So you put the slides, can you see the slides here? The slides are there, and then they are immersed in an alkaline treatment solution with that pH. After some time for the denaturation, like 20 minutes or something like that, you start the electrophoresis. Do you know what is an electrophoresis? Okay, which is the charge of the DNA? Negative. Okay, good. So let's imagine that the electrophoresis is going on, and then we don't have breaks in our nucleoids. So after the electrophoresis, okay, like nothing happened. There is no breaks. Let's imagine that we have few breaks. Then after the electrophoresis, the loops that contains those breaks are able to migrate. They relax. Remember that they are supercoiled, the nucleoids. So they will lose the supercoil, and they will be able to move and create a tail, small tail here. Now you see head and tails, okay? If we have a lot of breaks, the loops that contains those breaks will migrate. If we have a lot, then we will have big tails here. Yeah, okay? That's why they come. Yes, yeah, yes. No, make all the questions you need, please. So this is the main step, Okay, at this time, the comets are already formed. Why it is called Comet Assay is because they resemble to the comet in the sky. Okay, it's a fancy name. It has a boring name also, but we always use the Comet Assay. It has, how is the other one? Is cell gel electrophoresis, single cell gel electrophoresis assay. It's much nicer, Comet Assay. So, okay. At this point, the comets are formed. So now just what we need is to neutralize and washing. This is, well, during the neutralization, the hydrogen bonds will happen again if the bases are close. It helps with the staining. Then there is the washing, and then the staining. We use fluorochrons to stain the DNA, and then we visualize them under the microscope. Can you see it? Ah, well, no, not very well. Yeah. There is much nicer utility. So you can see the comets there. You can see comets with very small tails, and you can see comets with very big tails and very small heads. Right? Okay. So this is something I really want you to learn as a take-home message. What the principle of the assay is. There are DNA breaks. Those breaks will relax the loops, and those loops will migrate. Okay. And some people can ask, what about fragments? Well, there is still a discussion about it, but what we think is that there is a mix of loops and big fragments. Because if you have a very small fragment, probably you will lose it in the gel during the electrophoresis. Okay? A consideration, well, we are using a pH higher than 13 during the in the alkaline treatment and in the electrophoresis. And there are some kind of lesions called alkali labile sites. So these are DNA sites that will break in the presence of a high pH. And this is the case of the abasic sites, those places where the DNA has lost the base. When we have that in the DNA, when we have abasic sites, if we perform the assay, the abasic site will be converted into breaks. So we will also detect them. So what we can say is that the standard Comet assay detects DNA strand breaks and alkali labile sites. Okay, it's good to know these kind of things to interpret the results correctly. Okay, we have performed the assay, but we need to get the results. So normally, in these small gels, we have more than 1,000 or around 1,000 comets. What we do is we analyze 100 or 200, it depends, okay? But between 100 and 200 comets per sample. How we do that? Well, there are two ways of doing that. The first one is the visual scoring. We classify the comets in categories, but we do it visually. And then we calculate a comet score. And here, this is an example of 5 categories. So we are going to classify the comets that we see in our sample in class 0, when it doesn't have any tail. Class 1, it has a small tail. Class 2, it has a bigger tail, but same length of class 1, we could say. 3, it has like longer tail, and even the tail is a bit wider. And then class 4, it's very small head, and most of the DNA is in the tail. So you just classify yourself. I said, this is 1, this is 2, this is 1, 2, 3, blah, 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 blah. And when you stopped, when you get to 100, and then you calculate this comet score. Number of comets in class 0 multiplied times 0, plus number of comets in class 1 multiplied to 1, and the rest. So this comet score will go from 0 to 400. Okay? This is one way of doing it. Let's make an exercise. So, yes. Yeah. is a way of doing it. This is just an example. There are other ways. It's just to give a certain weight to certain type of comets. But you can make your own if it makes sense and use it. Okay. So maybe you look at the TV. Which class is this one? Look first at the screen and then at the TV, okay? 4, okay. This one? 3, 2, 3. Okay, this one?
1.
2, 1. This one? 1. 2. This one? 0, 2. Okay. Next. This one? This one? This one? Then this one? Okay. This one? 1, 2. This one?
2.
3. So I don't know if you have noticed it. You are all agree on 0 and 4, but the rest, yeah, it's a bit subjective, no? But it's working. It's really working. What you have to do is like the same person should score all the samples in one experiment or in one project. Okay, but we have another options. Use of image analysis software. In this case, software is going to calculate different parameters per comet. So I don't, but here, yeah, you can see here maybe. Calculates the head length, the tail length, the head intensity, the tail intensity, tail migration, tail moment. So it's different parameters. I'm not going to go into a lot of details, but the most used one is the tail intensity, which is the percentage of the DNA that is in the tail. So if a comet has a 0% of DNA in the tail, means that it doesn't have any damage. It doesn't have a tail. If it has 100% in the tail, it means that all the DNA is in the tail. Okay, and then you have everything in between those two values. So from 0 to 100. Okay, so then in this case, you also have to score or to analyze 100 or 200 comets, and then you calculate the mean or the median per sample, and this is the data you have per sample. There are two types of image analysis softwares: semi-automatic, in which the operator has to select the comets with the hand, and then the program is giving you the different values, and you stopped when you reach 100, 150, the number you want to score. But there are some that are automatic. This is you put the sample, it is a scan, and then it gives you the data. They are very expensive. I know Goran has one, right? Do? No. Send one to me. Okay. So, but this is the standard Comet Assay. So we are measuring only breaks. It's single-strand breaks, double-strand breaks, and alkali labile sites. But there are other versions. So we have versions to detect oxidase bases and alkylated bases. This is what we call enzyme-modified Comet Assay. Well, here is just these two examples, but this enzyme-modified Comet Assay can also detect uracil miscyl corporation, which is another lesion, or the IP sites we were talking about, the alkali labile sites. They can also be detected with this. As we are going to see an example, so you will see how it works. There are other versions like inter-strand cross-link Comet Assay. You can detect inter-strand cross-links. There are another version that use the Comet Assay in combination with DNA repair inhibitors. It has been used to detect bulky adits, but we are not sure if it is really can be used for the diagnosis of the bulky adults. In this case, what we do is to stop the repair. So the breaks are accumulated, because breaks can occur also in the repair. Yeah, so while the DNA is repaired, you can get breaks there. It's also something you have to take into account when you are interpreting the results. So there is a way to stop, and then the repair, and then you can, like, the breaks are increasing. Yeah, do you understand what yeah, okay. And then there are other versions to detect global methylations, which is an epigenetic event. It has not been validated also, but it works. And then DNA repair. You can measure DNA repair with the Comet Assay. And this is something Sabine Lange will talk about it tomorrow. Okay, let's see a bit of the enzyme-modified Comet Assay, because it's very used. Okay, the cross-link Comet Assay is not very used, and the DNA repair inhibitors is not very used yet, but this is really, really used, also in human biomonitoring and ecological biomonitoring. So this is the protocol that we have seen before. So we obtain the cells, we embed them in agarose, we lyse the cells, alkaline treatment, electrophoresis, neutralization, staining, and visualization. When we are going to do the enzyme-modified Comet Assay, sorry, I'm going to specifically explain the FPG-modified Comet Assay. So we are going to use the enzyme for amidopyrimidine DNA glycosylase to detect oxidized purines, mainly 8-oxoguanine. So this is bases that are oxidized. This enzyme is a bacterial enzyme that plays a role in the DNA repair in bacteria. So it's the one that detects the lesion and induces a break in the site of the lesion. Since we can detect breaks, we can detect those extra breaks that the enzyme is going to produce. So, okay, I will explain it more carefully. So in step two, when we embed the cells in the agarose, what we have to do is to prepare 3 identical slides instead of 1. And then we have an extra step between 3 and 4. So after the lysis, what we are going to do is to put FPG, the enzyme, in one of the slides. So you put a drop of FPG in one gel, a drop of FPG in the other gel. With other slide, you put FPG reaction buffer, so it's a control. Okay, you put a drop, and then we have an extra slide. Remember that we have 3 identical slides that we are going to keep in lysis to follow the standard Comet Assay. So with these 2, we will put them in an incubator for a certain time. And during this incubation, the FPG will introduce breaks at the sites of the oxidized purines. So let's imagine that, yeah. Okay, the FPG, the enzyme, need a buffer to give it the good conditions to work. No, the other way around. This is the FPG. Here you have the FPG in the buffer that needs to give the conditions. And here you have only the buffer without the FPG. Okay, it's a control. Yeah. So let's imagine we have a nucleoid with a lot of 8-oxoguanines. Okay, it has a lot. You do the standard Comet Assay, the standard one. You don't use any enzymes. What would you get? No tail at all. There's no breaks, no tail. There are no breaks, okay? No breaks, or let's imagine there are no alkali labile sites. So nothing. You can see, if you are using a compound, you can see this compound is not genotoxic. No. This compound doesn't induce breaks, okay? It may have other lesions that I'm not detecting. Now we will have this the same nucleoid with 8 oxoguanines, and we use FPG that will convert all these dots into breaks. What will you have? Tails, okay? A big tail because of the presence of 8 oxoguanines. Okay? Yeah? So let's see. We have one slide that we kept in lysis to follow the standard assay. What are we going to measure in this slide? What are we measuring with the standard Comet assay? Okay, so DNA strand breaks plus the alkali labile sites. Sometimes we forget about them, doesn't matter, okay? But it's plus the alkali labile sites. Okay, what we are going to measure in the second one, so it's the nucleoids incubated with the FPG reaction buffer, but without the FPG. Yeah, the buffer sometimes, you don't know why, induction breaks. Normally like a very small amount, but sometimes it happens. So, but we will also have this. Okay, they don't disappear. So in the buffer we have the DNA strand breaks plus the alkali labile sites that already have in the sample, plus the extra breaks or alkali labile sites induced by the FPG reaction buffer. What will we have? Would we have here? Yeah, perfect. We have these two plus the oxidized purines. So what do I have to do if I want to know only the level of oxidized purines in my sample? Subtract. I subtract this to this, and then I will have the net oxidized purines that I have in my buffer, or in my cells. So I have to, if I do both at the same time, which is very easy, if I do the standard Comet assay and the FPG modified Comet assay, I will have all the information about DNA strand breaks and alkali labile site, and also about the oxidized purines. Okay? Is it clear? Yeah? Okay. Some considerations. Assay conditions. One tricky thing about the Comet assay, I'm not going to go into details because we can spend here a couple of days, is that the assay conditions can really affect the results. Imagine that you put a very strong electrophoresis or a very long electrophoresis. What is going to happen with the tails? They are going to be very big. Or if you combine, remember that the cells are in an agarose matrix. The agarose can be very dense or can be totally the opposite. You can use 1% DNA, 1% agarose, you can use 0.4. The DNA will migrate easily if you put it in a 0.4% gel. Okay, so this is something to keep in mind, that the assay condition sometimes determine the results. That's why there are a lot of standardization going on. That's why we published this paper in Nature Protocol, just to try to make people use the same protocol so we can compare the results. Dead cells. What happened with the dead cells? How is the DNA in a dead cell? Apoptosis, for instance. How is the DNA in apoptosis? Very fragmented. So you have to be careful with that. In fact, if it is very fragmented, you will lose it during the electrophoresis because they are tiny, tiny pieces of DNA. But if you are in a very early stage of the apoptosis, you can have like class 4, let's say. Do you remember class 4 comets? Class 4 comets. You may think that is due to the compound that you are testing, but it may be due because of the apoptosis. So you have to check always in the Comet assay if the cells are dead or not. Always, okay? And then control or assay controls. You need to put controls. It's very easy when you are doing genotoxicity testing to treat your animals or your cells with a compound that you know that is going to induce the lesions you are measuring. In the case of human biomonitoring, that's not possible, but then you can play with assay controls. Assay controls are cells. It can be a cell line that you treat with a compound that you know that is going to induce the lesions that you want to check, and then you include it in the runs just to check that the electrophoresis and everything, like the whole protocol, went well. Okay, so it's not the proper positive control or negative control, but it's an assay control. Yeah. I'm going to finish with this. I told you, well, I have explained the Comet Assay with these slides where you can put two or three squares with the cells on it. But there are other versions, okay? This is the 12 mini gels. Remember that I told you at the beginning that you can have not millions, thousands of cells per slide, but then in fact you score only 100, 150 or 200. So you can use these mini gels in which you have around 300 comets, okay? And then you can increase the throughput of your assay. You can also use 12, 24, 48, 96 mini gels in gel bond films. It's something different to this, okay? It's like a plastic. You can also use the Comet chip, but I'm not going to go into details, but it's a chip of cells. So the cells are placed in a certain point. When we do the Comet assay with the gel bond or with the slides, the cells are like everywhere. They can overlap sometimes and makes the analysis a bit complicated. In this case, they follow an array. Okay. And then, of course, another way to increase the throughput is to use the vertical Comet assay. Normally, we do the electrophoresis in an horizontal way, but there are tanks that allow you to do it in a vertical way. That way, you can put a lot of more slides per tank. Well, here we have some graphs. Okay, this is HK2 cells. It's kidney cells. We treat them with ochratoxin A. It's a mycotoxin. It's a food contaminant. Okay, so we treat them. We use different concentrations. If you see an A, okay, sorry. So we have different concentrations of these mycotoxins. Time of treatment, if I remember correctly, is 3 hours of incubation. And this is our positive control. Hydrogen peroxide is a very good positive control. So it's induced a lot of breaks and it's not toxic to handle. So, well, ochratoxin is quite toxic. So what you can see here, this is the negative control. And this is the breaks. So is ochratoxin A inducing breaks? Do you want me to explain a bit more? So you have cells. Imagine like a 6-well plate. Have you ever seen it, a 6-well plate? Then you treat each of the wells with different concentrations of ochratoxin A. You have a negative control, of course. Here it is, negative control. And here a positive control. So a compound that we know it is going to induce breaks. And then we have these concentrations. And then you perform the assay. I have this. This is total Comet score. It's visual scoring. So what can you see here? It's similar to the negative control, right? So nothing. It doesn't induce breaks. Okay. Here we do the same, but with FPG, the enzyme we are talking about, and endo3 is another enzyme that detects other type of oxidase bases. is the same. You have the negative control, you have the positive control, and then you have, well, the positive control, I have to say, was not the best one. And here you have the different concentrations. Actually, the statistics are done, so you don't see and start. So what can you see? It's the same as the negative. So ochratoxin A in these conditions doesn't induce breaks or oxidase bases. Okay, let's see if I have one with. Okay, this one. Aflatoxin B1. Do you know aflatoxin B1? Yeah, it's another mycotoxin, very potent. It's carcinogenic for human, class 1 in the IARC. It's classified as class 1, so it's known that it's produce cancer in persons. Here we have, okay, let me remember, this is 3. Okay, this is very interesting because this is a cells, probably the same cells. No. I don't see the cell line, but it's probably the same one. Okay, anyway, a cell line. Yeah, that is what I was going to say. Yeah, here is just the cells are treated with aflatoxin B1. You can see, I mean, this is the angstrom breaks, nothing. Here is treated with, I mean, it's ochratoxin 1, but use the Comet Assay in combination with enzymes, and we don't have any oxidase lesions. But here, what we have added is S9. Do you know what is S9? Someone said here. Yeah, it's from liver. You take the liver from a rat, you smash it, and it's a way to mimic the metabolism that occurs in our liver. And there here, what you can see, there is an increase in the number of breaks, right? Nothing happened with the oxidase bases, but the breaks increases. It has a complicated explanation I'm not going to go through, but let's see. Okay, this is in vivo. These are rats treated with some nanoparticles that were produced to be a carrier of some medicaments. Okay, yeah. So it's for an oral ingestion. So, let's see this. We have performed the Comet Assay in the stomach of those rats, in the duodenum, in the jejunum, in the ileum, and in the colon. We have, this is the negative control, is the black one. The next one is 500 milligrams per kilogram. The other one is 1,000, and the other one is 2,000. And then we have the positive control, which is always okay. So what can you see here? Yeah, and you see here in duodenum, the high concentrations is inducing a lot of breaks. Okay, and here is the same, but with the FPG assay, and you can see exactly the same. Stomach is okay. Duodenum, here we have more than in the control, and then in the rest is the same. The error bars are different animals. Okay, in the case of the Comet Assay, you always use 5 animals. Something more. That's the last one. Okay, just the last one. Here we are testing if the. Okay, this is what we did is to take pepper. Yeah, pepper. Yes. Vegetable. So we put it raw, so without any kind of cooking, and we grill it. Okay, and then we digest them in vitro. Okay, with different enzymes and different treatments in terms of temperature, pH, and so on. We are recreating what is happening in our digestion. Okay, and then when we finish, we perform the Comet assay. If you look here, the strand breaks plus alkali labilisides, okay, the standard Comet assay, nothing happened compared with the control. Yeah, doesn't matter if it's raw or grilled, nothing happened. But when we try with the FPG Comet assay, we saw a clear increase in the oxidase bases produced by this food, even if it was eaten raw or grilled. We didn't have a clear explanation for that. But it was, in all cases, it was a non-toxic concentration, not only in the MTT, but also in the proliferation assay that goes a bit further. It was negative. So, yeah, I was involved in this paper, but it's from another group in my university, and I know they are continuing working on that. They are checking which compounds are produced here that could affect the DNA in that way. Okay. Sorry? It was HT29. They have a bit of metabolic, but not that much. But so maybe if you put the metabolic activation, these disappear. You never know. I mean, you have to take everything into account. The way of detecting crosslinks is a bit different because crosslink, they decrease the migration instead of increase the migration of the tail. And here you have this cisplatin and mitomycin C. They are both crosslinkers. And if you see, like if you treat cells with different concentration of these compounds, instead of having an increase of the damage or even like nothing, you see a decrease in the damage because they are producing these crosslinks. Here, vitamin CC is the same. But those compounds, they don't produce crosslinks, or they are not supposed to produce crosslinks, and then you have like nothing. So it's a way of detecting crosslinks.